Thank you very much, Ian. It's actually a pleasure to be here this afternoon. We were chatting earlier, and one of the interesting things about the sort of work that we do is I give seminars and talks to my own group within the centre, or else I give them to completely different groups way away from QUT. So rarely do I get the opportunity to really converse with our own community. So it's terrific to have that opportunity this afternoon. So what I'm going to be talking about is the world is getting hungrier and how can we respond? Well, in one word, urgently. We've got a huge amount to do. But importantly, why is the world getting hungrier? It's not that complex, and we actually know a lot about it already. We know, for instance, the population is growing. We all know the population is growing. That's very, very obvious. We move around the world. We move around our own cities. We see massive increases in population. The scary thing is by 2050, we're going to have another 2 billion people. So if you think that the population pressure already is pretty strong, by 2050 we're going to have another 2 billion people, and we're going to be up around about 9 billion. And this is nearly unavoidable unless we have a huge meteorite shower, or we have a Spanish flu-like epidemic but 100 times worse. We're probably going to get to that 9 billion people. So that's a lot of people. And most of that growth is in Africa and Asia. And that's where we have some of the biggest problems with food production. The other one of the other contributing factors, and it's one that people don't recognise very well, is a rising middle class. And rising middle class is very good. It means a lot of those countries that were mired in poverty for so long are now coming out of that. And they actually have significant disposable incomes now. So the first thing you do, if you're coming out of poverty, and you've got disposable income, is you want more food, you want better food. That's a very, very obvious outcome, and it's very well documented. But the interesting thing is there's a rapid increase in demand for animal protein. That's also okay, except that conversion of plants to animals is very, very inefficient. So you can feed a lot more people on a certain amount of plant food as opposed to a certain amount of animal food. So that's a huge problem, and so this is actually contributing to why the world is getting hungrier. The other big one, and we're all aware of it, and I'm sure hopefully all of us recognise it, is climate change. And there are a whole lot of components to that climate change that are going to affect food production. Certainly higher temperatures and reduced rainfall, we know about these, but it's really these ones that are going to have the biggest impact. The erratic weather conditions and also those extreme weather events. To give you some example, we do a lot of work in East Africa. East Africa, particularly in the highlands, absolutely fabulous climate. It's because it's on the equator, sun comes up at 6 o'clock in the morning, goes down 6 o'clock at night every day of the year. They've actually shifted it to 7 and 7, but that's mainly for tourist reasons. They also have a long wet season and a short wet season. And those wet seasons were absolutely predictable. Within 2 or 3 days, the long wet season would start. Within 2 or 3 days, the short wet season would start. The farmers could plant on a date. So this is date-based agriculture. Whatever the date was, we would plant our corn. We would know that it would mature in that period. That's changing, and that's changing quite dramatically. The day that the rains start are no longer predictable. The length of the wet seasons are no longer predictable. What we're now having is major crop failures in some of the areas that used to be incredibly stable. The other big one are these extreme weather events. We all know about drought. We all know about flooding. Massive impacts on food production. There's another one, though, that we don't recognise as readily, and most people don't, and that is that crops flower usually over a relatively small temperature range. That's their optimum. And if you go way over that or way under that, they don't flower very well, or they don't set seed. And what's happening now is when we get these extremes of temperature, over 40 degrees, if that's the time when that crop is flowering and setting seed, we'll get massive drop in In seed set, massive drop in yield. So this is actually, these ones are starting to have a really big impact already. So if we put all those together, it's not a particularly pretty picture, but even worse, so we've essentially got the perfect food insecurity storm. And it's on the way. We know it's coming. We actually know it's coming, which is a real concern, but it is a serious and global grand challenge. So we know that this is going to happen, so we know that the food and nutrition crisis is almost certain unless we do something really serious. By 2050, we will need to produce an extra 70% of food every year over and above what we produce now. Now that's a big ask, because at the moment we've got reducing water, we've got reducing inputs, and all of the other inputs are starting to be really significantly decreased, and yet we've got to increase food production by around about 70% per year. Obviously, an enormous part of that is to ameliorate climate change. That's difficult. It's both physically difficult, but particularly it is politically difficult. But we also need to vastly improve our agricultural production. So how can we go about that? What are the options? Well, certainly the sorts of things that contribute, much better distribution systems, particularly when we talk about Africa and we talk about Asia. The infrastructure in many of these countries is very poor. The transport systems are sometimes just dysfunctional, and a lot of that has to do with borders, where border crossings become extremely difficult. Logistics are a major problem, but also food storage. So there are significant advances we can make in this area, but it's certainly not the area that I'll be talking about today. Improved production systems, certainly. And they're going to be all of the different systems. It's not going to be limited to one system. In many parts of the world, and in Australia, organic farming is becoming increasingly popular. That's got to be one of the responses as well, because not only from a philosophical point of view, there are places in the world that don't have access to the sorts of inputs that we use that are dependent on organic farming systems. So that's going to be part of it, but also all the way up to large-scale industrial agriculture, precision farming. We've got a major program at QUT now around robotics, and that's going to be part of the solution as well. So all of these things, increasing the or improving the production system is tremendously important. But perhaps the major area is we're going to need different crop varieties. We're going to need crop varieties with much greater yields and improved nutrition. We can do that in two ways. One is by conventional breeding, and one is by genetic modification. Almost certainly, in the future, we'll be using a combination of both. One doesn't displace the other. They can be very, very complementary. The interesting thing is when you look at that, in our society particularly, we see organic farming and genetic modification as in conflict. In reality, they occur in two completely different segments of what we need to do. One's about production systems, and one's about generating new varieties. So I believe very strongly that we need both of these, and as well all of the other approaches as well. Conventional breeding, precision agriculture, etc. So if we start to look at new varieties, what are the sorts of things that we need to do? So one of the huge ones, and we're starting to work on it in our centre, is stress tolerance. How do we produce crops that will withstand drought for much longer, that can be submerged during flooding, that can withstand saline soils, but also can withstand those very high temperatures during the flowering process? These genes are already available for many of these characteristics. For instance, there's a gene that's available now in rice which allows the rice to be completely submerged for up to 3 weeks, which is fabulous, because that happens. At the moment, when rice is submerged for that long, it dies. But these genes have become available. Certainly pest and disease resistance, we lose a huge amount of production through pests and diseases. From a nutrient perspective, micronutrients, and I'll certainly be talking about this, increasing the amount of protein in our crops, but also digestibility, because a lot of the seed grains that we eat aren't particularly well digested by us. Many of them contain proteins, for instance, that are very good proteins, but they're just not digested. So certainly there are groups looking at how do we make our food more digestible. Very importantly now, with the genomics revolution, we're starting to see the sequences of virtually all the major crops coming through. We're now starting to identify all of these genes. We're now starting to identify the functions of all of these genes. We're now starting to identify how these different genes and different functions are passed on from one generation to another, which is really fabulous. So we're now able to look into these genomes and say, okay, I want to move that gene over there from that wild relative into here. I can do that by conventional breeding if the wild relative is closely related, or I can do it by genetic modification if they're not. So really what it's about is it's about improving our staple crops. So what are we doing at QUT? Well, let me start not by talking about plants, but by talking about nutrition. Micronutrient deficiencies. Look around the room, there's probably very little chance that anyone in this room suffers from any micronutrient deficiencies. In the Western world, it doesn't happen very often, and if it does, you go down to the chemist and you buy a pill. But around the world, micronutrient deficiencies are the third most important public health problem worldwide after HIV, AIDS and malaria. Those two swap around. If there are other mosquitoes around one year, malaria is higher. If they're not, HIV, AIDS is higher. And interestingly, the micronutrient deficiencies affect developing countries much more than anywhere else, but so do these other diseases as well. So these guys get hit really badly. The deficiencies, vitamin A deficiency, iron deficiency, anemia, zinc deficiency, folic acid deficiency and iodine deficiency. Some of these are relatively easy to solve. Iodine with iodized salt works really well. One of the interesting asides there is that many of us now are sort of working towards only having sea salt. Sea salt doesn't contain any iodine. There are small pockets of population which now have iodine deficiency because they don't have iodized salt. And that's in the Western world. Crazy stuff. Okay, let me talk about vitamin A deficiency, and this is the one I'm going to talk about primarily today. I won't talk about some of the other work we're doing. Map of the world, the red countries are where we've got clinical vitamin A deficiency. Vitamin A deficiency, what are the outcomes? Death is the extreme, blindness, night blindness. The two that have only recently been well documented is impaired immune systems and impaired brain development. If you live in the sorts of countries we're talking about, sub-Saharan Africa, I should say the blue bits are bits where there isn't data. So this is Democratic Republic of Congo. Almost certainly that should be red. So most of sub-Saharan Africa and most of South Asia have very high levels of clinical vitamin A deficiency. And most of those countries also need really good immune systems. That's where they're being challenged most commonly with some of the really serious infectious diseases. Okay, put some numbers around that. Around about 670,000 kids die every year from vitamin A deficiency. And around about 350,000 go permanently blind. So unbelievable outcomes every year for more than 1 million kids. That doesn't count the kids that have night blindness or lower levels such as reduced immunity or reduced brain development. These are just the real extremes. So the interesting thing is that we've had programs for a long time about reducing micronutrient deficiencies. However, it's still the third most important problem in the world. These are the strategies that have been used. Food aid, terrific, give people food, works for a very, very short period of time, is very expensive and is not sustainable. But people still put it on the list. These two have been incredibly successful, supplements and food fortification. They are expensive, but they are sustainable and they are being sustained around the world. But they're ongoing. Constantly got to go and provide these. However, despite these being around for probably the last 30, 40 years, there's still a population which is absolutely resistant to these strategies. And the reason for that is there's populations that don't buy food. They only eat their own food. So food fortification doesn't work. And those people, and we're really talking about the poorest of the poor, don't access health clinics very often. So the supplements don't work very well either. There had been, but this is really starting to drop off, a push to get these populations to change their diets and change their crops. We found out that this is pretty unsuccessful. If you're a subsistence farmer, you probably have a small plot of land on which you have to produce all your food. You've been growing rice for 10 generations, you've been growing bananas for 10 generations, you've been growing cassava for 10 generations, and then somebody comes along and says, don't do that, you should be growing this. They won't do it. They will not change their systems. So how can we get to this resistant population? Well, what we're doing now is what's known as biofortification of staple crops. That's taking the food, taking the crops that these people naturally eat, and increasing the amount of micronutrients in them. And this is becoming a very strong program around the world, both by conventional breeding and also by genetic modification. Conventional breeding, if the genes for those high levels of micronutrients are available and we can move them over by conventional breeding, best way to do it, because it's so much quicker to get through from a deregulation perspective. But in many instances, those genes aren't available in an accessible gene pool. So we've got to move them a different way, and the way we move those can be by genetic modification. Okay, going back to staple crops, what are they? These are the top 10 crops in the world. Everyone here will recognise the top 4, but maybe not all of these ones, particularly as staple foods. Rarely in Australia do you see cassava or sorghum or yams in the market for human consumption. But look how important they are. They're all, and particularly cassava. Soybeans, not so much a staple food. We all see bananas, plantains and sweet potato in the markets, but not as staple crops, more as a little add-on. But these are staple crops. And if you look at the ones that are now being biofortified, and this is by both conventional means but also by genetic modification, 6 of the 10 crops are now in advanced stages of biofortification, particularly for what's called provitamin A, and I'll talk about that in a moment. So very much a target for increasing the provitamin A levels in these staple crops. From a genetic engineering point of view, this is the most advanced. This is golden rice. Polished rice doesn't contain any provitamin A at all. I should say provitamin A is a combination of alpha and beta carotene. Alpha and beta carotene are orange, and that's why it's called golden rice. This was developed around about 10 years ago. This is normal polished rice. White, this is the first version of golden rice. This is golden rice 1. And this is golden rice 2, which was developed probably around about 8 years ago. And it has more than 30 micrograms per gram of beta-carotene equivalents. That's how we measure it. It's in advanced stages of development. It's gone through multiple field trials, and almost certainly it's going to be released in 2014 in the Philippines. It's already been demonstrated in a number of human feeding trials just how efficacious it is to be able to have this as part of your diet and within the amount of rice that you would eat every day. There was concern that the colour would have a reaction, or the population would have a reaction to the colour. That hasn't been so. That seems to not be a major problem. But at QUT, we're targeting bananas. And many people don't recognise, but plantains, which are grown particularly in West Africa and a cooking banana, they're just a subset of bananas. So talking about bananas, and they can be both plantains and bananas. Very importantly, in 2005, we were really privileged to receive a grant from the Bill and Melinda Gates Foundation under their Grand Challenges in Global Health. Bill Gates came up with, obviously with help, 14 grand challenges in global health. Most of those were around vaccines, antibiotics, control of insect vectors, etc. But one of them, grand challenges number 9, was creating staple crops with a complete set of micronutrients. And under that, they funded 4 projects, ours, which is Banana 21, Bananas for the 21st Century, cassava, rice and sorghum. So all of these crops clearly in the top 10 of crops, staple crops in the world. A little bit about bananas. I'm sure all of you are used to eating dessert bananas. This is rather different. So bananas, top 10 crops in the world. It's the largest fruit crop in both volume and value. In the West, it's a dessert fruit. But in developing countries, particularly in the wet tropics, it's a really key component of diets. And I often give people a little challenge. When you see film of a developing country in the wet tropics, have a look in the background and you will always see bananas growing, or nearly always. Incredibly common. Around about 100 million tonnes per year, a third in Africa, Asia and the Americas. 15% is the world export market, and that's primarily Cavendish, the bananas that we eat. 85% consumed and traded locally. So by far the vast amount of these bananas are being consumed, normally very, very close to production. The highest consumption in is Africa's Great Lakes region and in Papua New Guinea, and consumption in some areas is as high as 1 kilo per person per day. Breakfast, lunch and dinner is bananas. They're triploids. There's a little bit of science. They're triploids from 2 wild progenitors, Musa acuminata and Musa balbisiana. Interestingly, where they come from, the centre of origin of bananas is from the tip of Cape York. So if you go up to Cape York, you can actually find wild seeded bananas all the way up through Indonesia, all the way up through Papua New Guinea, Malaysia, and across Southeast Asia, South China, and then right across to South Asia. So most people think of Latin America as the home of bananas. No, they're all imported from our region. They're herbs, so they're a giant herb. They don't have any woody stem at all. They're parthenocarpic, very important. They produce fruit with flesh but no seeds. Very important. They're perennial. They're propagated with suckers, but very importantly, they fruit all year round in the tropics. If there is sufficient water over the entire season, bananas will produce a crop. So they're now becoming a really important food security crop. Where the rainfall is no longer predictable, you know that the banana won't die. It can go up to 8 months without water, and once the water comes, it'll produce a crop. So it's becoming an increasingly important food security crop. Just a quick one, this is what they look like, and diagrammatically, here's the bunch. They have a corm underground, they have a sucker that comes off the side. They produce 1 bunch per plant. When this is produced a bunch, this is cut down, and this grows up and takes its place. That's the sucker, and it becomes what's known as the return crop. But bananas have a huge problem. And one of the problems why genetic modification is important. The vast majority of domesticated bananas are selections from the wild. They were never bred. So Cavendish that you eat today, 2 stories about Cavendish. One was it was collected from a backyard in Saigon, and the other one was it was collected in southern China. Somehow it made its way over to the UK, I think Kew Gardens, where clearly its name was changed. Cavendish doesn't sound like either a Chinese name or a Vietnamese name. So we suspect the name was changed. All of those bananas, all of the ones we eat and many others, are just selections from the wild over many thousands of years. And they're sterile. So when you cut a banana down the middle, sometimes you see little black specks. They're the aborted seeds. So, unlike nearly all other major crops, bananas have not been genetically improved for thousands of years. The way to look at that is that they haven't had sex for a couple of thousand years. So it may be one of those very frustrated crops. Because they're sterile, they're exceptionally difficult to breed conventionally. You can do it, but you've got to go back to the wild relatives. You've got to recreate the bananas that we grow today. And that's an extremely long and extremely difficult process. You can't take a Cavendish banana and improve it. It just doesn't work. And also, Most of the conventional breeding programs, and there are quite a few in the world, are completely focused on finding a replacement to Cavendish. That doesn't really help those people that eat bananas as a staple crop. Okay, East Africa, this is where our work outside Australia has been concentrated for a number of years. This is around the Great Lakes region, Uganda, Rwanda, Burundi, Tanzania, Kenya and the Democratic Republic of Congo. Uganda has been our major target as the collaborator in this project, even though those other countries are very important and will become more important. When you look at the numbers, and this is from a 2006 survey, somewhere between 15 and 30% of the kids under 5 had clinical vitamin A deficiency. Around about the same number of women, and when we go to iron deficiency anemia, the numbers are even worse. They go up to 80% for kids and 65% for women. The small number is in Uganda, is in Kampala, where there certainly is access to all of those supplements. The big numbers are out in the regions. And the staple food of Uganda is bananas. And these kids, a photo that I took, almost certainly would have vitamin A deficiency. A number of you probably donate clothes to various organisations, so this is where those clothes end up. But I will tell you, every now and then you have a funny story, and I think Anthony James is in the audience here somewhere. He and I were on a field trip in Uganda, and we were in we came into a small and very poor village, and a girl probably in her mid-20s was on one of the little plantations we went to, and she had a Wallabies jersey on. And Anthony said, "Oh, you're a Wallabies fan." She looked at us as if we were absolutely crazy. It's interesting, the cultural divide. Just to give you a sort of taste of what it looks like, these are very, very common images all around Uganda and most of that Great Lakes region. People carry 3 bunches of bananas on a motorbike. This guy's probably done his weekly shopping. Women without push bikes will carry bunches on their head. This is how they're cooked. So this is the standard mud hut that you'll see in these very poor communities. This is over a fire, a pot with boiling water in it. The bananas are peeled, cut, wrapped in banana leaves and steamed for a number of hours. So this is a traditional cooking method in Uganda, and different variations, all based around steaming, are also around that region. So our project is not about genetic modification of bananas. Our project is actually around alleviating micronutrient deficiencies, particularly vitamin A deficiency and iron deficiency in Uganda. And that's through micronutrient enhancement of the staple food of Uganda, which is bananas. They have a number of different types of bananas in Uganda. The East African Highland banana is the cooking banana. That's the one that predominates. But they also have small sweet bananas. The Cavendish is a beautiful small sweet banana. And Ndizi means banana in Swahili, and Sakali is sugar, so it's a sugar banana. And they also make beer out of bananas. I can absolutely guarantee this is not going to be a winner internationally. I actually really enjoy beer, but this is one that just completely beat me. But around that region, Rwanda, Burundi, Democratic Republic of Congo, Tanzania and Kenya, also very high levels of vitamin A deficiency, of banana consumption, and also vitamin A deficiency. In West Africa, they grow plantains. These are also essentially cooking bananas. And the project is a collaboration between QUT and the National Agricultural Organisation of Uganda. Okay, a little bit of background. Provitamin A is a combination of alpha and beta carotene. These carotenoids are converted to vitamin A or retinol in the human liver. So that's very important. And very importantly, humans only convert the amount of provitamin A to vitamin A that is required. This is really important because it means people can't overdose. You can have buckets of beta-carotene and we'll have no negative health outcomes at all. Might go slightly orange, but certainly no negative health outcomes. Vitamin A, on the other hand, which we could engineer into bananas, does have a higher level toxicity. So that's why we've deliberately gone for alpha and beta-carotene. And they're orange coloured, as we saw with golden rice. We've got a target of 20 micrograms per gram dry weight beta-carotene equivalent. So this is our target, and it's important to remember, 20 micrograms per gram. And we're going for 2 farmer-preferred varieties, the East African Highland bananas, but also a new banana called Hybrid M9, which is very much like the East African Highland banana, but has quite a high level of disease resistance. So it's potentially a very important banana for that region. So how do we arrive at this number? How do we get to 20 micrograms per gram? Well, we know, and it's published, the estimated data requirement for vitamin A and for both children and adults. We had to work out what the conversion was of these carotenoids to vitamin A. That was very important because the matrix, whether it's bananas or rice or whatever, will affect how easily that provitamin A is converted to vitamin A. And surprisingly, this was done in an animal model in the very well-known Mongolian gerbils. We've never heard of Mongolian gerbils before, but apparently Mongolian gerbils have a system of converting provitamin A to vitamin A very similar to humans. I think there's only one person in the world that does that, and that's Sherry Unpronounceable second name over at the University of Wisconsin. And so we shipped bananas over to the University of Wisconsin. She did the feeding trials on the Mongolian gerbils, and bingo, we came up with a much better number than we thought we were going to use, and we actually used around about 6 to 1. Daily consumption, those figures are available. We then had to work out how much of these alpha and beta carotene were lost during the cooking. So our lab and the lab in Naro turned into a kitchen, and we steamed the bananas and we measured it, and luckily around about 75% of the carotenoids are retained during steaming. So that's how we came up with this magic number of 20 micrograms per gram. Okay, importantly, bananas, unlike rice, do contain some alpha and beta carotene. In fact, some bananas contain huge amounts. This banana, Asapina from Papua New Guinea, you remember our target is 20 micrograms per gram. This contains nearly 140 micrograms. And I should say this is a mature, green mature, what we call full green. Cooking bananas are harvested mature but green, and that's before any of the starch has been converted into sugars. So they want the starch, they don't want the sugar. So Asapina, massive amounts of beta-carotene. The bananas that we have, Cavendish and Ladyfinger, very, very poor. East African Highland bananas is around about halfway to our target, and Hybrid M9 is about quarter of the way towards our target. So we knew where our starting point was. All of the work that we did in Australia was with Cavendish. We've done a little bit with Ladyfinger. This is a complex project. We started right at the very, very beginning of the project. We said, we're not going to produce the bananas in Australia. They're going to be grown in Uganda. The Ugandans are. We're going to develop the technology, and we would transfer the technology on a continual basis to Uganda, and they would do their own genetic modification. So at the end of the day, the bananas that came out at the end would be made in Uganda. But very importantly, we do everything first in Australia. And that goes for deregulation, field trials, feeding trials, whatever, will be done outside Uganda before taking it into Uganda. This is so a little bit of science. I'll throw a little bit of science in. This is the pathway for producing alpha and beta carotene. So various compounds and various enzymes. Golden rice increased the amount of this enzyme, or switched it on, and put another enzyme in that replaced these two enzymes here. This is an important compound here, geranyl diphosphate. It's essentially a crossroads molecule. So a lot of other things hang off that. It doesn't just go to make alpha and beta carotene, it goes to make plant hormones and everything else. So one has to be very delicate in how we manipulate this pathway to dramatically increase the amount of these compounds without affecting anything else. So we've got to be very careful that we don't affect anything else. A whole lot of technical challenges when we started, transformations, which genes we're going to use, which promoters, how we're going to analyse these, and how it's going to regulate and cope with it all. So, this is how we did it to start with. We took the rice strategy, which was a gene out of maize and a gene out of a bacteria called Erwinia, and put it in. We also went and we took this wonderful banana from Papua New Guinea that has this incredibly high level of provitamin A. We took the genes out of that. And so that's been the basis of the project. One of the problems with bananas is they're slow. So this is months down here, where if we start a project, we have to produce what we call embryogenic cell suspensions. These are essentially stem cells. So these are cells, single cells, that are omnipotent. They can grow back to a whole banana. Single cell, whole banana. We can genetically modify the single cell and regenerate a complete banana from that. But making these is not an easy task. And we've got particularly one person in the lab. It's more of an art than a science. And whenever Jen goes on holidays or something similar, the whole process falls apart. But we're getting better at it. So 9 months to get our cell suspensions, 12 months from transformation to rooted plantlets, 6 months to plants ready to go in the field, 12 months to grow those plants, and 6 months to harvest them and analyse them. So that is around about 4 years. So this is a long-term commitment, and if your funding is only for 4 years, you've got to do something pretty dramatic to make sure you get all your milestones. So what we did is we recognised that bananas were too big to assess to fruiting in the glasshouse. The time scale was too long for iterative or serial experimentation. So the only practical alternative was that we were going to go directly to the field. We were going to take every permutation and combination we could possibly think of, bung them in the field and see what works. Amazing, and we called this massively parallel field evaluation. Apparently somebody else has used massively parallel before. Ours mightn't be quite as big, but certainly it's been a fabulous strategy. The field trial we put was up in Innisfail. Unfortunately, it's in the cyclone belt, and we did get hit by a cyclone. Just south of Innisfail, we had this field trial. That's what it looks like. So the bananas, and it's quite large, it's about a hectare of bananas. We've had around about 1,300 different transgenic lines going through that field trial. And in the most part, these bananas look perfectly normal. So we tried 3 genes, the banana gene, the maize gene, and the bacterial gene in various combinations, and we drove that by what's known as different promoters, and these are the on-off switches. So these two here are on-off switches for fruit, and this one is a switch that's on all the time, and so is this one. So we're interested to see which one was going to work. Very depressing to start with. So this is the golden rice strategy. So we copied exactly what they did. Seems a pretty smart thing to do. Remembering our target was 20 micrograms per gram. These are a whole lot of different lines down here. The best one we got was 3.5 micrograms, way under our target. We'd still increased by more than 2.5-fold, which is pretty impressive, but we were way under our target. When we substituted that maize gene that was in the golden rice with the banana gene, we did better. And we got up to 6 micrograms per gram, which was nearly a 6-fold increase. So we were getting better, but still way under target. However, then we went to a different promoter, got rid of the bacterial gene, and bingo, way up, very close to our target. And this was a number of years ago, so we're pretty excited about this. And the wonderful thing was, yes, they're orange. So this is Cavendish. These were once they're ripened. This was Cavendish. This is one of our bananas, a much more rich and orange colour than the wild type banana here. The very good thing is that many of those bananas had huge bunches. The plants looked normal. We were getting, and this is probably our favourite photo of normal bananas. This is normal Cavendish ones, and this is probably one of our best lines here. So these bananas have fabulously rich golden flesh, and we were very excited. What was even better though was when we had a look at the ratoon crop, and this is the crop that comes up. Now, those first figures were from the plant crop. We put the plant in the ground, it produces a bunch, then we let the sucker come away and we analysed the bunch from those. And bingo, we were way above target. In fact, one of the lines had double the amount of beta-carotene equivalents, provitamin A, that we needed. This is the one we're most interested in, so it's well over target. But we've got a number of other lines coming through as well. And this is Cavendish. This is going from a very, very low base. So way over target. Things are looking fabulous. We've got a way to go forward. We also started field trials in 2010 in Uganda. If you're ever travelling out of Kampala on the Bombo Road, I suspect there aren't too many people here that will be, you'll be able to look out the left-hand side about an hour or 2 hours outside Kampala and see a QUT sign up. Not exact. I'm glad Peter Colbrack's not here. Not perfectly painted, but pretty good. So this was when we planted the trial. This is Jeffrey Aranekwe. He's our senior scientist on the project in Uganda. This is the field trial. This is the first one we had coming through. This is one of these Scullendisi. And you can see, yes, there's an increase in the orange colour, but it's not a dramatically increased level. Very importantly, though, that this field trial was probably the first field trial of any crop, any genetically modified crop in sub-Saharan Africa, where the events were made in a national lab, in a national African lab. Virtually all of the other events have been made usually overseas or by some of the international organisations. So this was really important event, and the guys have done unbelievably well. We came back from Uganda a couple of weeks ago, and we now have a line over there, and this is our hybrid M9. So this is our target. This is one of our targets. We now have a line with 45 micrograms per gram of beta-carotene, so more than double what we want. So this is a fabulous outcome that we only found out about 3 weeks ago. And this is in a field trial in Uganda. Okay, so what are the major outcomes out of there? We've got lines with greater than target levels of provitamin A in Australia. We've got one line with greater than target levels of provitamin A in Uganda. The best gene is a banana gene, and that's the gene out of this little banana from Papua New Guinea, Asapina, that we've moved across. Works really well. And very importantly, the high PVA bananas generated in Uganda by Ugandans for Ugandans in Ugandan varieties. We're now going into development phase, and that's important. It's a much more complex phase rather than the wonderful research concept, the proof of concept phase. Just checking my time. That's going reasonably well. One of the other area that I wanted to talk about, and this was the front page of, or the cover of New Scientist in 2003, predicting that by 2030, now, bananas will be extinct. So, killed by the plague, which was very good. So what is that plague? Well, it's actually a number of plagues. One is a disease, and they're all diseases. One is a disease called black sigatoka, it's a fungus. Another one, Fusarium wilt, which is a fungus, occurs in 2 types. The other one is a virus called banana bunchy type virus. In South and Central America, Cavendish is very susceptible to this disease. Luckily, it doesn't occur in Australia at the moment. They are spraying 70 times a year. A third of the total cost of production is the cost of fungicide. And this is so people in Europe and North America and even New Zealand can get bananas that are absolutely pristine. all of the residue is left back in Central and South America. It's not sustainable. This is a disease called Fusarium wilt, tropical race 4. It kills, and it kills Cavendish. And this is bunchy top, and this was a photo that, again, Anthony and I took. This is in the highlands of Rwanda. This was an important one because it was the first record of bunchy top in East African highland bananas in the highlands, so that was a real concern. Okay, we've got a program directed towards Fusarium wilt resistance and banana bunchy top resistance, again using genetic modification. The targets now are both Australia, Africa and Asia. So rather than just concentrating on one area in Africa, we're much more broadly focused. This is race 1 of Fusarium, and this is where it occurs, the red blotches. That's essentially where bananas are grown. So this is where they originated. We've carted bananas over here and over here and over here. We've also carted the diseases. Humans are wonderful at moving diseases around. The diseases themselves, the fungi and the bananas, had really good systems for moving around until they discovered humans and thought, wow, this is a much easier way. This is the worst one. Tropical race 4 of the same fungus. It occurs in one area in Darwin, in Indonesia, Malaysia, in Philippines and in China, and it's spreading quite rapidly. It's the really big challenge because it kills Cavendish and virtually all other banana cultivars. This is a soil-borne fungus. There are no fungicide controls for it, and it stays in the soil for at least 40 years. So it's a massive problem. This is what it looks like. In Australia it occurs in the Northern Territory, but not in Queensland and New South Wales. This is what it looks like. This is inside a banana that has succumbed. That's this banana here, and when you cut it open, that's what it looks like. This is the last remaining plantation in the Northern Territory. We work on this plantation. It's the only plantation that produces bananas for the Northern Territory. What you'll see here is bananas. This is quite a young plantation. This is probably 12 to 18 months old. You'll see holes, and you'll also see yellow. The yellow are the plants that are already infected. The holes are the ones that are already died. So this will last. The grower up there replants every second year. Normally bananas are replanted every 10 to 15 years. He has to replant every second year. And he's only just holding on. So what we've done is we've developed a strategy to modify these bananas. We've modified Ladyfinger for resistance to race 1 and 4 because it's susceptible to race 1. We've modified Cavendish for resistance to race 4. We've used resistance genes from a wild banana. So this is a seeded banana called Malacensis, which comes from Indonesia. And we've used what's known as stress tolerance genes from rice. If you look at what happens to Ladyfinger bananas in the glass house, this is what we call our small plant test. Here's a plant before we've infected it, and progressively, once we put the fungus in the soil here, progressively you see this plant die. And over about a 2 to 3 month period, we go from that to that. So it's a pretty dramatic death. However, we've now got plants that don't do that. So these are ladyfingers. They're genetically modified with a stress tolerance gene out of, I think this one's out of rice. So here's our control. This is our normal ladyfinger banana that's been inoculated. This is our control banana, which is ladyfinger but hasn't been inoculated. And here are the transgenic plants. So these plants have been transformed with a stress tolerance gene, and they look great. And even when you open them up, they've got a very, very high level of resistance. Not quite immunity, but a very high level of resistance. But this is the one we're really interested in. This is tropical race 4. As I say, we've got a field trial now up in Darwin at that same property that I showed you a little while ago. We've got Cavendish and Ladyfinger, both with stress tolerance genes. But importantly, we've got Cavendish with a resistance genes out of this wild banana, Musa acuminata malacensis, which is really interesting. It's a seeded banana. Here we are in Brisbane. This is our field trial for provitamin A. and this is where we are up in the Northern Territory, about an hour's drive from Darwin. This is what the trial looks like. We always have to have a sign up to warn people about the dangers of these bananas. Essentially, it's keeping people out. This is not an area people go to. When we were setting up the field trial, the trial manager said, "Well, we only have a few things that we need to be careful about. Just watch out for the buffalo, because they're quite nasty." There was a girl taken by a crocodile a couple of months ago, about 2 kilometres up the road. The snakes are a bit thick, and we've got to worry about the dingoes. So I thought, geez, this is one of the best places you could possibly do a field trial. I think I saw Stephen Pincus up there somewhere wondering about the risks of doing field trials up in the Northern Territory. This is what the field trial looks like, but very importantly, and these are photos that just came back last week, this is one of the bananas that we've got a banana resistance gene in, and it's looking fabulous as compared to the susceptible. We're not quite there yet, we're very close, but we're very excited about starting to get these genes coming through, because again, they're banana genes. Bunchy top has been a disease that we've been working on for a very long time. It's transmitted by aphids and in suckers, and once the plant becomes infected, you get complete yield loss. So it moves around very easily. And it's moved around the world. So it was first recorded in Fiji. That probably wasn't where it originated, but that was where it was first recorded. 1890 in Taiwan, Egypt in 1901, in Australia in 1913. So this was really interesting because it's part of our heritage. By 1920, the number of plantations in southern Queensland and northern New South Wales had gone from over 500 down to 4 because of this virus disease. When you drive around Queensland, particularly as you drive up north, you'll see signs up, prohibited to move bananas. The reason is this disease, bunchy top. And one of the ways they controlled it was to have incredible legislation around controlling how particularly banana suckers moved. So banana inspectors can go on to, and if you look at the Queensland legislation, it's amazing, can go on to any property at any time and amazingly confiscate any material. It doesn't actually have to be bananas. It's incredibly strong legislation. But it's worked, and we haven't eradicated the disease, but we've certainly controlled it. However, in Africa, it's been a different story, and it's getting worse and worse and worse. This is a really nice series of slides that Anthony James put together. This is where it was first recorded in Africa in 1901. And I'll just flick through this and just watch how the grey increases over time. So Democratic Republic of Congo, then it moved into Central African Republic and Gabon, then it moved into Zambia and Malawi, and then it moved into Angola and Mozambique, and now it's moved into Nigeria and Benin, and it's getting worse. And I think the picture that says it all is this one. And this was a photo we took in Malawi, and this is not unusual. So this is a banana plantation that has come infected with bunchy top. This would have been all bananas. Not one of every single banana in that plantation is now infected, and they will not produce any fruit whatsoever. And obviously we're working towards transgenic resistance for that virus. So bananas, QUT and food security. Bananas, top 10 crop, staple and the most important fruit. The first GM, so our accomplishments have been first, the first GM banana field trials in Australia, provitamin A enhanced bananas in the field in Australia and Uganda. We've now got fusarium wilt resistance in bananas in Australia, and we'll soon be taking that elsewhere. And we've got bunchy top resistant bananas coming through. Just some key points in our target countries in Africa are Uganda, Kenya, Rwanda, Tanzania, Burundi and the Congo. Ultimately, we will be working on plantains as part of this and move into West Africa. And we've already started in Asia and India, In India in 2012, funded by the Indian government, and we'll probably start in Indonesia in 2014. In Africa, our target population for the micronutrient enhanced bananas is 125 million people. We were amazed when we actually worked out what this number was, but it's a huge number. Our release date for the high provitamin A bananas in Africa is 2019-2020. And very much the project has been around collaboration, technology transfer and education. These have been really key components. These are the students from East Africa that have been involved in the project and enrolled at QUT. The ones in bold are students that have already completed their PhDs, and these are students that are currently enrolled. So this is a very important part of the project, very important part of QUT's contribution. And I think there are 11 students that have been involved, which is fabulous. But we've got some fabulous supporters. We were very lucky about in December 2011, Bill and Melinda Gates were in Sydney on holidays, and they took the time to fly up to Cairns to visit the project. They did come up in their private jet, so that was a little bit easier than the average person. But what was amazing is that a number of us, so Harjeet, myself, Rob, Mumbi and Joive, met with Bill and Melinda for 3 hours in the afternoon. I think the thing that struck us, I mean, apart from being totally surreal, was their absolute interest in the project. Bill had a pile of papers about this high that we'd sent. He'd read them. He was asking questions which were as good a questions as you'd expect from a scientist who was involved in the area, but not absolutely. His questions were very insightful, and it was just amazing. And he took, 20 pages of handwritten notes. Melinda, on the other hand, also absolutely involved in the project and understanding the project, but very much from a social perspective. What impact was that going to have on the families, in particular on the women? In Africa, most of the farming is done by the women. So she was very interested in how those women were involved in banana production and also whether we were involving them in the project. So it was just a fabulous opportunity to meet two people who are really making a difference. It was just wonderful. Okay, finished with huge numbers of people involved in these projects. So I just get to come and talk about them, about the project. These are the people at QUT that are involved. These are the people in Uganda that are involved at the present time. We've got field trials in Innisfail with Jeff Daniels and in Northern Territory with Mark Smith. Clearly, support from the Bill and Melinda Gates Foundation. We were funded for phase 3 last year, and that'll take us through to the end of 2015, which is then the final push to releasing the product. So the wonderful thing about this foundation is that they're patient donors. If I went to somebody and said in 2005, I'm going to develop this wonderful banana and I'm going to have it ready in 2020, you don't get too many people willing to stand up and say, yeah, we'll fund that. So that's the sort of difference having these types, or particularly this group. We've had funding from the Australian Research Council, which is great, and particularly the field trial up in Northern Territory, Lamana Bananas. Also, I've got to thank our own university. QUT's been fabulous in providing opportunities for students to come here, fee waivers, etc., as well as putting up with me doing the project. And I'm going to leave it there. Thank you very much.